All right, welcome to part 3 of the notes for cellular respiration, where we're going to talk about fermentation, which we saved till last, and so actually I guess the better title of this unit would be cellular respiration and fermentation. I want to go back to the cellular respiration parts, which we've already covered in part 1 for glycolysis, parts 2A and 2B, where we talked about Krebs cycle and electron transport chain. And in objective 5 here, you can see we're going to talk about how many ATP are being made, just again to kind of sum this up, but also to kind of compare how many ATP are being made in cellular respiration versus fermentation, and you'll see it's quite a bit different. So across the top here under number 5, make sure you have these things written down: glycolysis, Krebs cycle, if you want to abbreviate, ETC, and then our total. And then in there, if you want to also use some mathematical symbols. We're going to add all of these up, total ATP, so we can start to make a little bit more sense out of this unit. So again, for glycolysis, which actually produces 4 ATP, but uses 2 of them to complete its process, so we say a net gain here of 2 ATP. Oops. And then in Krebs cycle, we don't have anything too complicated going on here. Also producing 2 ATP. So you can see between just the first 2 parts of cellular respiration, not a whole lot of energy going on. When we get to electron transport chain, that's when we start talking about big numbers. And so why electron transport chain is so important, why oxygen is so important, because without oxygen, neither of these two take place. Glycolysis can take place with or without oxygen, but Krebs cycle and ETC are the respiration parts that make it respiration. We're talking about these guys taking place only if oxygen is present. So, obviously our totals. For cellular respiration are 36 ATP. So let's get into talking about fermentation. And very simple, we're just going to start off with the purpose. The purpose of fermentation is actually to provide glycolysis with more NAD plus in order for glycolysis to actually keep making ATP. Again, glycolysis is making 2 ATPs for each glucose molecule. Fermentation doesn't necessarily make ATP, but it does make NAD plus, or return NAD plus back to glycolysis so it can continue making more ATP, and that's the most important part. So here's a quick definition of fermentation, or not. It is an anaerobic process that takes place in some bacteria, yeast, and muscle cells just like ours. So mammal muscle cells. And again, we want to make sure we remember what this is talking about when it says an anaerobic process. And something else to keep in mind, we're going to be talking about muscle cells for part of this. So really quickly, let's get into this discussion of anaerobic versus aerobic. Anaerobic respiration refers to the creation of energy in the form of ATP without oxygen being present. So I guess this would be the fermentation. Aerobic refers to the creation of energy in the form of ATP when there is oxygen present. So something you may want to add to your notes here. We're basically, for this part, talking about fermentation. And here, we are talking about simply respiration. meaning Krebs and ETC. If you want to fit that underneath, that'd be fine too. So without oxygen, organisms rely on fermentation. With oxygen, the preferred method with oxygen after glycolysis, the preferred method is Krebs cycle and ETC. Again, going back to these numbers. 36 ATP for 1 glucose is quite a big deal. So number 7, we're going to take a look at specifically lactic acid fermentation and alcoholic fermentation. And also talk about just some general fermentation stuff here too. So just to go through the in general stuff, we have glycolysis taking place before fermentation. Keep in mind this is before fermentation. The molecule that goes into glycolysis, recall from part 1 of the notes, that sugar that a lot of plants are making. Thank you, plants, for your glucose. Ose, sugar. So glucose is going to go into glycolysis here. But that's not all that's going in. As I briefly mentioned earlier, we also have NAD+, that's going to go into glycolysis. And then, of course, the energy parts that we have to talk about, because that's what this unit is all about. ADP and a phosphate group are going to get added. And glycolysis is going to turn around and spit some things out, and in this case, for fermentation. So let's go back to our glucose molecule. Again, this is our 6-carbon molecule. And really what happens to glucose is pretty much it just gets split in half. So glycolysis is going to split glucose into, that was supposed to be a 2, 2 pyruvates. We've already seen this when we talked about Krebs cycle, but glycolysis isn't doing anything different here. And again, those pyruvates are 3-carbon molecules. So 1 6-carbon into 2 3-carbons. Should be no surprise there. NAD+ goes in. It's going to pick up electrons and a hydrogen and come out as NAD. I'm putting a dot dot to represent those electrons, NADH. And if ADP and a phosphate go in, hopefully obviously by now, ATP, triphosphate, is going to come out. So there's the basics of glycolysis, and how it's important to fermentation is down here at the bottom. The pyruvates are going to go in and actually become other things that we're not going to talk about. But what happens is these electrons right here get donated to the pyruvates during fermentation. So this is going to get sent back to glycolysis as NAD+. Again, glycolysis is using NAD+ to make its energy, ATP. It's going to actually get rid of electrons there, put them onto NADH. NADH is going to give its electrons to pyruvates, and these electrons keep cycling back and forth from glycolysis to fermentation. And then NAD+ will come back, pick up more electrons, and keep going. So this is kind of how fermentation provides energy, not directly, but through the process of glycolysis, providing glycolysis with the stuff so that it continue, because NAD+ gets used up pretty quickly. Alright, next up is lactic acid fermentation and kind of its basic parts. It takes place in some bacteria in our muscle cells. Uses NADH, again electron carrier, dot dot, and pyruvates from glycolysis. You just saw that up here in the diagram. Creates NAD+ to supply glycolysis, again, so that more ATP are being made. That's the goal here, to produce more ATP. It also produces, hopefully, obviously, lactic acid. and is used in industry to create things like yogurt and other dairy products. So pause the video if you can't keep up with that, and we're moving on to alcoholic fermentation next. So alcoholic fermentation takes place in yeast. It also uses NADH and pyruvates from glycolysis, no surprise there. Also creates NAD+ to supply glycolysis. Here's the difference in alcoholic fermentation. There's also carbon dioxide being produced. And again, hopefully, obviously, alcohol. And in this case, used in industry to make things like bread, wine, cheese, beer, and things along those lines. So again, keep in mind that if there is not oxygen present, something like your muscle cells will proceed with fermentation, lactic acid fermentation. It's a good reason that why if you were to try walking home from school you could do it, but sprinting home you can't because your body is using up too much oxygen than it is replacing. And in that case, your muscle cells start to get sore. There's a buildup of lactic acid there. And the chemistry there is going to make you feel it that night or the next morning. So that's part 3 of the notes. I hope you like